हाँ सर, शुरू कर दीजिए।
Webinar is now streamed. Okay, good afternoon, everybody. We have this afternoon session. Dr. Deepak Singh Bist is a scientist at the National Institute for Plant Biotechnology, IARI. He will be talking about the techniques used in molecular biology, basically the PCR and Southern, and also gene annotations. It's a bunch of words, phrases rather, uh, which he will try to cover in the next 1 hour. Deepak is a student of IRI. In fact, he is my student. Now he's a scientist, and he has a keen interest in teaching. In fact, in the past workshops, I think he was the darling of all the participants. And we look forward to a very interesting presentation and a lucid presentation from Deepak. In fact, what I have been telling all the speakers is that at the end of the lecture, at least leave a couple of questions for homework, because as teachers, you are all always familiar with homework. Now that your positions are changed, now you are students, I want that the speaker should give some homework to all of them. With these words, now I hand over to Deepak for the lecture. Deepak, take over.
Okay. Thank you, sir. I think my voice is audible. Just एक बार confirm कर लीजिए sir. आप interrupt कर सकते हैं whenever there is some connection is not there. So
No, it's audible. Go ahead.
okay. Thanks. So good afternoon to all of you, and thank you, sir, for introducing me or the people who are in this meeting room. So I was given a task here to talk about polymerase chain reaction, hybridization techniques, Sanger sequence, and sequence annotation. And exactly these terms are also there in the textbook in Unit 5 of Class 12. So that's why we thought that we should clear all this, the concepts of all these techniques, so that you can disseminate the knowledge with whatever you are gaining here to your students or in your respective schools, from your respective schools. So before I venture into detail of all these things, I just want you people to have a brief idea of what exactly DNA is. So DNA is basically a double-stranded structure, and there are 4 different building blocks of DNA: adenine, thymine, guanine, and cytosine. So these are building blocks, and without fail, this adenine will pair with thymine, and the cytosine will pair with guanine. So this you have to remember throughout this lecture, that this pairing is very important. Adenine will always pair with thymine, and cytosine will always pair with guanine. There is no other possible combination. And there sometimes we have some exceptional bases also, but they are of no use in this lecture. So we'll skip all those things. We'll focus on all these 4 important bases and how they pair. So this is the snippet of what I have taken from your textbook. This is basically PCR. It is given in Unit 5, page number 17. So a representation is being made here, a small cartoon, I think. So in this case, what they have done, they have provided you with 2 different strands of DNA. Then a simple step is being shown here: annealing, extension, and all those things. And this is what is written for PCR. So I'll just try to make it more easy to understand. So I made a specific cartoon for all of you, although I'm sorry for that it not look so good, but I tried in by a simple paint software in my system. Technology, I can say that it is basically foundation of molecular biology. Without PCR, no lab can exist. A molecular biology lab can't exist without PCR. So what exactly in PCR, what we are doing, we are making multiple copies of a specific fragment of DNA. Just to make you clear here, we are not amplifying the entire genome. Genome is basically the complete set of DNA in a particular cell, a haploid set basically to be more specific. But we are not amplifying the entire genome. We are interested in only in a specific fragment of your DNA that can be amplified in PCR. Your interest here is to make thousands and thousands and copies of that specific fragment. These copies are made for some specific purposes. You might go for cloning, you might go for sequencing. So whatever you want to do, you may have to make multiple copies of a particular fragment, then only you can proceed for your downstream experiments. So again, I'll mention here that in PCR, what you find this specific. So the first and foremost, for amplification, what we need to denature your DNA. Denaturation means you have to separate your two different strands. Then only new strands can be synthesized. If you don't separate your strands, DNA strands, new strands can't be synthesized. Again, just to tell you, all these steps are also going inside your cell. Also, right now, also they are going inside your body cell, but they are going at an ambient temperature, say 37 degrees centigrade. And more importantly, inside your cell, there are several other proteins also, which play a very significant role in stabilizing all this mechanism which is going inside. And you cannot mimic the entire thing outside. So denaturation also happen in a normal cell, but in a normal cell, there are specific proteins like you have this helicases and single-stranded binding proteins and all those things which stabilize your DNA. But in outside environment, to exactly mimic that thing, it is not at all possible. So in this case, what we do for denaturing your DNA, denaturation, what essentially is done here is we give a high temperature treatment to your DNA. So above 70 degrees centigrade, your DNA will start unwinding. So normally what we do in lab, we keep it for 94 degrees centigrade. So at 94 degrees centigrade, we are assured that both strands of DNA will unwind. So this is needed because only on these unwinded strands, new strands can be synthesized. So first step is denaturation. And then in previous slide from that book also, you can see in this case, it is written denaturation step at 80 degrees centigrade, you are denaturing your DNA. Normally we do at 94 degrees centigrade. Now, the second most important thing is you need annealing. Annealing is basically, as I told you, you cannot amplify the entire genome. You have to, only a specific fragment can be amplified. So how the specific fragment can be amplified, that is the most important thing. So for amplification of that specific fragment, you need some primer. So since the sequence is known, without sequence information, you cannot design primer. So a sequence is known, and we wanted to know, so you can design primers. So these primers, what basically they are doing, they are delineating the region that has to be amplified. The primer will go and bind to the specific region of DNA that has to be amplified. So in this case, for your for simplicity also, and to make you understand, because sometimes it becomes very difficult to understand all these things. So I have given a sequence also here. I hope you might be able to see it. I have given this DNA sequence double-stranded. I haven't shown the bonding between this double-stranded DNA, but this green and this red strands are basically the double-stranded DNA. So if you have this sequence with you, so what from this sequence you can easily design primer. Say for instance, in this case, I have designed a forward primer, which is so for time being, you forget this red color sequence. And anyway, in database also, you only have this single strand sequence. No database have the sequence of a complementary strand, but you can easily make it complementary by the rule I told you at the beginning of this presentation, that A will pair with and G will pair with C. So wherever A is there, you can easily put T, and G is there, you can put C, and vice versa also. So for time being, you forget this red strand, you focus on this green strand. So if you have a strand information with you, you can design primers from that strand information. How it can be done? Say if I want to design a forward primer, you exactly take the sequence and that is I have underlined here. Exactly take this sequence. Say forward sequence is CTCAG. So in this case, I am designing a primer of say 5 base pair line, a reverse primer. Okay. I hope I am audible, sir.
I can't hear you, Deepak.
Sir, मैं आपसे पूछ रहा हूँ, am I audible to you? मैंने
No,
pause-
no. Now you are audible.
It's okay.
sir,
In
मैंने
between, your voice is breaking.
Your voice is breaking, yes.
सर उस समय तो मैं कुछ बोल नहीं रहा था, मैं आपसे पूछ रहा था कि ऑडिबल है कि नहीं. अभी है ऑडिबल सर?
Go ahead.
ठीक है सर. So we have this sequence with us. So if you wanted to design primer, so what we do basically is you take this sequence exactly as such, this forward sequence as such, CTCAG, and the reverse sequence is basically the complement of this. So in this case, the complement
बीच में ब्रेक हो रहा है.
if you focus on these end 5 bases, so the complement
Voice is not audible. Deepak voice आ नहीं रहा. No, I can't hear.
Deepak, your connectivity is very poor.
and I just want to mention
NRCPB का गड़बड़ है यार आपका.
am I audible now sir?
You are audible.
अभी है सर मेरा वॉइस? क्या देख सकता हूं मैं, बट आया था कि यहाँ पे कनेक्शन होगा. सर इफ यू कैन गिव मी 5 minutes so I can check in someone's lab, कहीं पे आ रहा होगा क्योंकि
नहीं, वही मैं कह रहा था कि because there is always a problem there.
so sir 5 minutes मैं करूं, ट्राई करूं सर एक बार?
Okay, you can, क्योंकि
I will pause for 5 minutes sir. बस 5 minutes मैं भी ट्राई करता हूं सर इसको.
कहाँ से करोगे? NRCPB का कहीं से भी होगा तो प्रॉब्लम होगा आपको.
तो मैं अपने घर से कर लेता हूँ सर, मेरे पास वाईफाई है, लाइन है हाई कनेक्टिविटी का. 5 मिनट लगेगा पहुंचने में सर. मैं इसी वजह से यहाँ पे आया था.
अच्छा, नहीं वहाँ बिल्डिंग के अंदर सिग्नल आएगा नहीं ना, आपको कुछ भी होगा तो. That's the problem. बिल्डिंग इज द प्रॉब्लम.
सर फिर 5 मिनट में मैं घर पहुंच जाता हूँ, वहां से कर लेता हूँ. बस 5 मिनट
ठीक
लगेगा
है,
यहाँ से पहुंचने.
जल्दी निकलिए तुरंत.
ठीक है सर.
तब तक भट्ट साहब will engage the audience.
ठीक है सर. ठीक है.
तब भी चलेगा, पता नहीं. बर्मन साहब
सर,
अभी तो आप आए. ओके, लेट I anticipated some problem with this one. I request everybody to bear with us for this interruption. We will continue